he was performing a surgery on an ectopic pregnancy. And he took the mass out and was thinking to himself, why are we throwing this away? There's got to be amazing stem cells in here. But to avoid all of the ethical issues that human embryonic stem cells went through, what he ended up doing was scrape off the pre-placental tissue from the outside of it. He handed the rest of the mass back to the pathologist. And the reason why they can throw it away is because in normal pregnancy, OBGYNs understand it to be non-viable. Well, my father, having taken the pre-placental tissue off the outside of the embryo, now has the earliest stem cells that could possibly source without any ethical issue.
What if the key to reversing biological aging was hiding in tissue that medicine routinely discards? My guest today has spent more than 2 decades pursuing exactly that question. Yuta Lee is the founder and CEO of Accelerated Bio, and he's built a platform around human trophoblast stem cells, the earliest ethically sourced cells from the embryonic stage. Today, we explore the biology, the ethics, and the manufacturing logic behind a cell therapy platform targeting age-related decline. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Yuta. It's good to have you on today.
David, happy to be here.
Yuta, share something that you believe about bioprocess development that most people disagree with.
Ooh, that's a good one already. Hitting with the hard questions. Most people disagree with. I think that only people in the industry know how difficult it is to manufacture biology or anything biology. And even if you set all of the conditions in the most ideal setting, the ideal SOPs, the ideal conditions, things will still go wrong. And I think that it's almost a myth that a lot of people who are not in manufacturing or in processing or process development, they believe that once you set the process, it's already done. And it's not. A lot of things can go wrong. And I think that the devil really is in the details. I think that it would be great to have everybody who's not in the manufacturing world, the CMC world, or the process development world to understand that this is a really difficult job because we barely know biology and we're trying to control it. And so it's very, very hard to do.
You're making a great point, and thank you for the reminder. And this is something I've tried to repeat as much as possible to all the people I speak to. Manufacturing and development are hard, very difficult. And it's actually surprising that you're making this statement because you came into biotech with a business background. So tell us your story. What sparked your interest in stem cells, and what were some of the pivotal and defining moments along the way that led you now to being a CEO?
Okay, I'm going to start with the story. Actually, the story starts with my father, who is an MD PhD. His name is Professor Jiaonan Lee, and he got his MD from Tohoku University in Sendai, Japan, and his PhD at St. Bart's at University of London. So I actually spent 4 years of my childhood in London, which was a lot of fun. Then he came back to Taiwan and to practice, and he became a key opinion leader for OBGYN. And he sent us off to Los Angeles, and that's why I have this very American accent. But what happened was in 2003, he was performing a procedure called a salpingectomy for an ectopic pregnancy. And ectopic pregnancy happens when the embryo is traveling down a fallopian tube. And sometimes, at usually 1% to 2% of the times, that embryo may get stuck. And if it gets stuck, it'll sit there and grow until it bursts the tube. And if it bursts the tube, the mother may die of internal bleeding. So it's actually the highest cause of death in first trimester pregnancies. So to make the long story very short, he was performing a surgery on a salpingectomy or on an ectopic pregnancy. And he took the mass out and was thinking to himself, why are we throwing this away? There's got to be amazing stem cells in here. But to avoid all of the ethical issues that human embryonic stem cells went through, what he ended up doing was scrape off the pre-placental tissue from the outside of it. He handed the rest of the mass back to the pathologist as a second to double check. Yep, it's an ectopic and they can throw it away. And the reason why they can throw it away is because in normal pregnancy, the embryo needs to implant into the mother typically by day 7 or day 8. But you don't discover the stuck ectopic pregnancy until about 4 to 8 weeks. So there are many weeks in between where that embryo is not connected to the mother. There's no nutrients coming to it. So all OBGYNs understand it to be non-viable. It's already gone. And that is the reason why when you perform this surgery, all you have to do is take it out, give it to a pathologist to double check, and they can throw it away. Well, my father, having taken the pre-placental tissue off the outside of the embryo, now has the earliest stem cells that could possibly source without any ethical issue. And he comes to me and he goes, 2 years later, so he discovers this in 2003. In 2005, he comes to me and goes, Yuta, you're the business guy. Do something with this. To which I respond, hey, Dad, I went to Berkeley for economics and law. What do you want me to do? That's pretty hilarious. As a good oldest son in the family, I was like, all right, challenge taken. I will take it on and I will see what I can do with it. So luckily, my brother-in-law was an investment banker at Morgan Stanley in San Francisco for healthcare. And so I called him up and I said, Bob, who's your go-to IP lawyer for this kind of stuff? And he says, oh, you got to go to Wilson Sonsini in Palo Alto, right next to Stanford, probably the best lawyers in town for this. So we go, we find a partner, and we file the first patent. And that was the beginning of it all. And lo and behold, 5 years later, we get our first composition patent. And it's so powerful that one of the top attorneys calls me and tells me, Yuta, your patent portfolio or your first composition patent is so powerful because you sit between all of the immortal cell lines, the human embryonic stem cells and the induced pluripotent stem cells or iPSCs, and the non-scalable MSCs or mesenchymal stromal cells. And you have the best advantage of both. So keep building out the portfolio. Don't tell anybody you have this because you're too early. This was 2010. And 2010, no one wanted to invest in stem cells. So all the VCs would not be interested. So keep building a portfolio. But most importantly, do not give your cells to any researchers because once they take it and publish on it, then all information is public, right? And if the schools are smart, Stanford will have a patent, Harvard will have a patent, and you have a thicket of patents above your head that if you want to ever take the cell to commercialization, you would have to license all of those patents back to have a pathway. I was like, wow, that is such an amazing piece of advice. So we went back and I talked to my parents and we ended up funding the first 10 years of patent prosecution out of our family estate. And so that's the reason why we now have the earliest stem cells that could possibly source without any ethical issues. And not only that, we've already taken it to GMP, which I'll talk about in a little bit. And it's got amazing characteristics, which I'll also go over in a little bit as well. But that's the beginning of the story. So that was 2005 to 2010, and we've been working at it ever since. I formed a company, Accelerated Biosciences, in 2013. And only in 2020, when the topic of allogeneic therapies became popular, that I came out and started telling everybody about it. So that's the origin story in a very, very long format.
What a fascinating story, and it's definitely a non-linear story. Really great. Tell us, Yuta, a bit more about stem cells. You mentioned different stem cell, different types, different stages. Let's start very simple. What are the different types of stem cells and what is important to watch out for?
I love that question because this is what most people want to know, right? So I usually explain stem cells in the context of fetal development. First of all, let's go back, take it one more step back. So what is a stem cell? A stem cell is the most original cell where we all came from. Every cell in our body came from that one original cell. You may already know that every cell in your body in the nucleus has your DNA and your DNA only. And so the original cell, the embryo that starts dividing, turns into a human being, all came from that same DNA. A stem cell is defined as a cell that can generate exactly a stem cell and also derive into a sister cell or a differentiated cell at the same time. And so it has multifunction, but it is the original cell. And so, relative to where stem cells come from, I usually use a timeline of fetal development. The first 8 weeks of our lives is the embryonic period. So that's when the sperm and the egg get together. And 8 weeks later, it will start to differentiate into different cell types in our body. And that period is called the fetal period, where our organs are developing. And then after that, we're all born on average at the 38th week. That's called a birth event. So the baby comes out, the placenta comes out, the umbilical cord, etc. And anything after that's considered adult. Where all stem cells come from, the earliest stem cells that you can find from the very, very beginning are embryos from an IVF clinic or in vitro fertilization clinic. This is for couples that cannot have children and they want to have children. So what they do is they take the sperm and the mother's egg and fertilize it into a dish and then create healthy embryos that they can now re-implant into the mother and hopefully she'll get pregnant that way. And when they create these embryos, they actually create a lot of them. And so, but they pick the healthiest ones to implant into the mother and the rest goes into a bank. And so in 1998, a professor named Jamie Thompson out of Wisconsin published the first paper on human embryonic stem cells. And what he did was he was able to get consent from one of the mothers to take a leftover embryo and start doing embryonic research. But that created a lot of ethical issues. A lot of people believe that those embryos could have been alive and therefore you should not touch them or destroy them or do research on them. And that was the beginning of the US government kind of shutting down any embryonic stage research because of ethical reasons. So that's the earliest that you can find. And usually you get these embryos at like 5 days. Next is ours. So our human trophoblast stem cells are discovered between 4 to 8 weeks post-fertilization. So still in the embryonic period, but it's trophoblast. So it's pre-placental. So if you remember my earlier story, when some of these embryos get stuck in a tube and we have to surgically remove it, we just take the pre-placental tissue from the outside of it, hand the rest of the material embryo back to the pathology, checks and throws it away. And so we have the earliest stem cells that you can find without any ethical issues. Anything after that from the fetal period, so about from 8 weeks all the way to the 38th week is the fetal period. If you get cells from there, you're getting it from an abortion. Okay, so no one wants to touch that really. And at the birth event, at 38 weeks on average, that's when the baby comes out. And when the baby comes out, a lot of things also come out with it. The placenta, the umbilical cord, the cord blood, amnion fluid. So there's a lot to be collected there, but that's much later stage. And those cells tend to be mesenchymal stromal cells or MSCs. And then anything after that is considered adult. So we actually have stem cells literally buried in every cell type in our body. So when you cut yourself and start bleeding, stem cells will come and regenerate the right cell types to heal that cut. If you damage a muscle, there will be stem cells inside your muscle that will help it heal and regenerate that muscle. So it really exists everywhere in our body. But as an adult, it's actually difficult to get. That's why most adult stem cells come from things like adipose or fat. So human beings have a lot of fat these days. So you can actually isolate that. Or you can get it from bone marrow, for example. But that's very difficult. That means you have to drill into the bone and suck the marrow out and then process it for stem cells. The last one I want to mention is induced pluripotent stem cells or iPSC. And for most people that have heard me speak before, I work in longevity. And these induced pluripotent stem cells, or iPSCs, was published by a professor from Kyoto University named Shinya Yamanaka. And what he did in 2006 was he published on using a Sendai virus and changing 4 transcription factors and setting that virus onto an adult skin cell and was able to drive that cell all the way back to a stem cell state. And the whole world was like, oh my God, what is this? He actually received a Nobel Prize in 2012 for that work. But what's really, really critical for our longevity or even health span is that if you're able to turn one adult cell all the way back to its original stem cell state, literally at like a factory reset, that means theoretically you can turn every cell in your body and reprogram it to an earlier state, right? So that means anti-aging or longevity is really just an engineering problem that we're all working on. So there are companies out there like Life Biosciences that you may have heard of from David Sinclair, also Retro Biosciences with Joe Betts-LaCroix. They're all using this reprogramming method to try and drive back, reprogram cells to an earlier state. So that's very, very exciting stuff that's happening right now in longevity. But quick recap then, where all the stem cells come from. Human embryonic stem cells, the earliest that you can find. The human trophoblast stem cells, the earliest you can find without any ethical issues. So these two are the only ones from the embryonic period. And then you can find cells from the fetal period, but they're from abortion, so no one wants to use that. And then you get a lot of MSCs from the birth event at 38 weeks when the baby comes out. You can get a lot of MSCs from adult cells drawn from our body. And then you have the iPSCs, which is any cell that you can take and you reprogram it back to the factory state. I hope that helps.
So that means that today, most of the stem cells that are being used clinically are from adult sources, right? That's the large majority today.
That's right. Most of the research is either using MSCs or mesenchymal stromal cells or iPSCs, which are the induced pluripotent stem cells. And what you do is you set it back to the stem cell state, and then you have to differentiate forward for any specific types of cells, make it into a neuron, for example, make it into a kidney cell or a liver cell.
Tell us, Yuta, now why you are focusing on much earlier cells. What are the advantages of these cells?
Right. In that timeline I just mapped out, the general rule is that the earlier you find the cells, the more biologically active they tend to be. These human embryonic stem cells, for example, are immortal. Literally, you can keep growing it, keep growing it, keep growing it. It'll keep growing until, of course, it starts to mutate or something goes wrong. Our cells are also very, very early, but they're not immortal. They're naturally senescent. That means that after a certain amount of dividing or growing, it will stop and it will die. But since we get it from such an early state, we're able to drive the population doubling out to 85 population doublings. Now, let me explain that to you. A population doubling is when a cell divides and doubles. So 1 to 2, 2 to 4, 4 to 8, et cetera, except 85 times. And at 85 times, that is equivalent to 8 to the e to the 25 cells. If you can imagine the number 8 and 25 zeros behind it, that's how many cells you can get from one donor.
That's a lot of cells. So tell us how many that is.
I don't even know what that number is, but I'll tell you it in context, okay? So on average, human beings have about 37 trillion cells. So if you're able to take that 825 zeros divided by 37 trillion, you'll end up with more than 2 trillion human beings, all from one donor. So scale solved. And that's why you want to go with earlier cells. But even with earlier cells, like the human embryonic stem cell, which is immortal, the FDA and many regulatory bodies are actually afraid to use it. I mean, outside of the ethical issues, they believe that immortal cells tend to have also immortal signaling. So if you think about it, what else is immortal in your body, right? Cancer cells. Cancer cells also don't stop dividing. They also keep growing until they continue to mutate and literally take over your body. So this is why when you take an immortal cell and you put it in your body, it tends to be a little more dangerous. And so we have to be very careful about how we manufacture that. So our cells, we don't have that problem. They're naturally senescing cells, so you know it will stop dividing. It will end at some point, but it is so high in population doubling that you get all the scale that you need. In contrast, an MSC that you find at birth event, maybe if you're lucky, you get 25 to maybe 30 population doublings without engineering it. And that's all you get. And the problem with that is you will run out of cells. or we have very limited doses to actually treat patients after you get through all of the clinical trials. And every time you go back and resource a new cell, a new MSC, for example, it's a new DNA. So the FDA will say, well, do it all over again, right? So this is the problem with scale and later cells. So our cells have scale. The pre-placental tissue, one of the main advantages from these human trophoblast stem cells, or HTSCs, is that these cells are pre-placental. And all placental tissue express a surface marker called HLA-G, or human leukocyte antigen G. So let me try and explain this another way. An embryo itself is half the father's gene and half the mother's gene. So technically, the embryo is a different DNA construct than the mother. So how come then this embryo can attach itself to the mother without her immune system saying, what is this? Get it off of me, right? So it turns out that placenta has that role to mediate everything between the embryo and the mother. And the placenta, using HLA-G, will downregulate all of the mother's immune system, literally saying, everybody relax, this thing's okay, let it attach, let it take nutrients from you, don't worry about it, okay? Which means that these placental cells have very natural immune privilege. And it's very important for the embryo to stay attached and survive and get nutrients and grow into a baby. And this HLA-G is so powerful that now you can actually take an embryo and implant it in the surrogate mother to carry the baby for you to term. So literally a woman that is biologically not even related to the baby can help carry that baby to term until its birth, which is amazing. But what a shortcut that we've discovered, right? So our cells have this. So if you combine those two characteristics, one, they're naturally senescing cells without any ethical issues. You can grow so much of it that everybody can use it. Two, they have natural immune privilege. That means everybody can use it now without any rejection. That's the perfect combination.
And I think this will open the way also to allogeneic off-the-shelf cell therapy treatments, correct?
That's right. That's what we're aiming for. And so this is also the reason why, even when I started officially launched a company in 2013, I didn't really come out and tell anybody about it until about 2020, because before then it was all autologous, because CAR T therapies, everybody was focusing on investing in autologous therapies. But now we have quite a complete allogeneic platform. We want to tell everybody about this. And so if you're a researcher or if you're a company that's thinking about creating an allogeneic therapy, call me. We'd love to talk to you.
We've covered a lot of ground today, from the biology of trophoblast stem cells to the IP fundamentals that underpin Accelerated Bio's platform. In part 2, we go further into the science of aging itself and what a therapeutic approach to health span extension might actually look like in practice. If this episode added value, please leave a review on Apple Podcasts or your preferred platform. And thank you so much for tuning in. I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.